Chapter 4, Section 5. Let's do this, guys. Let's talk about the endomembrane system. It is a series of intercellular membranes that compartmentalizes the cell. It breaks it down into specialized chunks. Restrict enzymatic reactions to specific compartments within the cell, so they know that certain things are only happening in certain places. This endomembrane system consists of the nuclear envelope, the membranes of the endoplasmic reticulum, the Golgi apparatus, and vesicles. There are several types of vesicles, and their job is to transport materials between organelles of the same system, so between parts of the endomembrane system. Let's start by talking about the endoplasmic reticulum. It is often shortened to the ER because that's just a mouthful. A system of membrane channels and saccules, flattened vesicles, which remember are just sacs of membrane material that are continuous with the outer membrane of the nuclear envelope. So it's hard to tell where the nuclear envelope ends and the endoplasmic reticulum begins. There are two different types of the endoplasmic reticulum, the rough and the smooth. The rough is studded with ribosomes. Protein anabolism, they synthesize proteins there. The endoplasmic reticulum takes those proteins that are made by the ribosomes that are stuck to it and modifies them and processes them. Oftentimes it adds a sugar to that protein, which then results in something called glycoproteins, and those are very important to cell functions. The rough ER also forms transport vesicles that contain those proteins so that they can be moved to the Golgi apparatus. The smooth ER does not have ribosomes, and its job is to make lipids. In the testes of males, that is where testosterone is produced. The ER is what does that, so in the testicles of a male, you would expect to see cells with a lot of smooth endoplasmic reticulum. They're the site of various synthetic processes, detoxification and storage. The liver, with abundant smooth ER, also detoxifies drugs. The smooth ER also forms transport vesicles that can move the substances that are produced in the smooth ER, can take those lipids and move them to the Golgi apparatus. So here is a diagram. Okay, you'll see here that the ER is studded with ribosomes, so we know that this is rough endoplasmic reticulum. And here, there are no ribosomes, so this has to be smooth endoplasmic reticulum. The Golgi apparatus, it was named for a guy named Camillo Golgi. It consists of flattened, curved saccules, kind of resembles a stack of hollow pancakes. And it modifies proteins and lipids with signal sequences. So it receives vesicles from the ER on the cis or the interface. It modifies them and prepares them for shipment and packages those proteins and lipids into vesicles that then leave the Golgi apparatus from the trans or the outer side. Some are transported to locations within the cell, and some of them are exported from the cell. Others return to the ER or are merged with the plasma membrane. Here we have a diagram. All right, so here is a transport vesicle. It is joining with the cis or the interface of the Golgi apparatus. Okay, it passes through. Over here on the trans side of the Golgi apparatus, you can see a vacuole that is being formed right here and one that has just been released. And it may travel to the cell membrane out here, over here, and whenever it joins with it, it's made of phospholipids, and it just kind of fuses with it, and if it is trying to expel, if it's trying to get rid of what's inside that vesicle, the membrane will kind of open up and allow the substances to flow out. Down here we have an actual image of a Golgi apparatus. So you can see it does look like curved pancakes. Lysosomes, they are membrane-bound vesicles that are not found in plant cells. They are produced by the Golgi apparatus, and they contain powerful digestive enzymes that are super acidic. and they are used to digest large molecules into smaller pieces that can then be used again. In white blood cells, the lysosome engulfs the pathogen, engulfs the bad thing, and basically dissolve it using that acid. Lysosomal storage diseases, there are some out there. They are caused by a defect in some enzymes that are found in those lysosomes. They result in a disease sometimes called Tay-Sachs, which is a developmental disease, and eventually all of the people affected with Tay-Sachs die. They don't live a very long time. We can talk more about it if you guys are interested in class. Scientists are working on gene therapy to restore the enzyme that is missing in those lysosomes to help those individuals live longer. Here we have an image of a lysosome. Okay, right here we're going to blow it up. Here is the lysosome, and it has engulfed this mitochondria that is no longer functioning correctly, and a peroxisome. Okay, and it is going to digest these two structures, break it down into smaller pieces so that they can be used again in the cell. So here is a summary of the endomembrane system. Proteins produced in the rough ER and lipids from the smooth ER are carried in vesicles to the Golgi apparatus. The Golgi apparatus modifies these products and then sorts and packages them into vesicles that go to various cell destinations. Secretory vesicles carry products to the membrane where exocytosis produces secretions. Exocytosis is simply, it's like cell pooping almost. It's where the cell just sends stuff out to get rid of it. Okay, so mammary glands producing milk would be an example of this as well. Lysosomes fuse with incoming vesicles and digest macromolecules. So here we have a diagram, our nucleus with the nuclear envelope with the ER fused to it is creating vesicles, the smooth and the rough, the smooth over here, the rough over here, sends them to the Golgi apparatus where they are processed and shipped out. You can see the lysosome over here doing some digesting. Okay. Cells can also take in materials the same way that they secrete them. A vesicle will fuse with the membrane, like right here, and it will fuse with the membrane, and once the cell's like, yep, this is good stuff, it opens up and allows it to move into the cell. So that is it for the endomembrane system. If you have any questions, shoot me an email, guys.